Dear students, I suggest you to watch a short clip on somaclonal variation.
The genetic changes or the variations that are observed in the plants regenerated through the in vitro cultures of somatic cells are called as somatic variations. Though these variations have been observed in the in vitro cultures for a long time, but these were usually ignored, thinking these are abnormalities and are not useful. However, in early 1970s, their potential for the agronomic improvement was realized, and since then lot of progress have been made to understand the biology of the somaclonal variations and their commercial use for agronomic betterment. The term somaclones was coined for the first time by Larkin and Scowcroft in 1981, where soma means the vegetative and clone means the identical copies. That is, the variations which arise in the vegetative clones of the plants are called as the somaclonal variations. Other terms have also been used to describe such variations like the gametoclones, caliclones, or protoclones, or mericlones. These names have been derived from the sources or the origin of these clones. Like the clones that are derived from the culture of gametic cells, that is the microspore culture or anther culture, these are called as the gametoclones. And the genetic variations observed in these plants regenerated from microspores or anthers are called as the gametoclonal variations. Similarly, caliclones is the term given for the callus cultures. Protoclones is the term given for the protoplast culture, and mericlone is for the meristem culture. And the variations observed in these cultures are respectively called as the caliclonal variations, protoclonal variation, or the mericlonal variations. Depending upon the objective of the experiment, somaclonal variations may be desirable or undesirable. For example, if the objective of the in vitro culture is the clonal multiplication of the elite cultivar, like what we earlier studied in the micropropagation, these variations are undesirable, as the introduction of these variations in the elite cultivar can result in the loss of the quality or the quantity of the commercial product, or it may introduce other unintended characteristics. On the other hand, these variations can be an important source for the genetic variability and can be used to either improve the existing crops or to develop the new desirable or novel genotypes that may have agronomic and commercial advantages. These variations may be caused by several factors and can be temporary or permanent, like the changes in the tissue culture raised plants that result from the physiological or epigenetic effects usually do not inherit to the next generation and are reversible, and therefore these variations or these phenotypes which results from the epigenetic changes or the physiological changes, these are temporary ones. Since their identification in 1970s, somaclonal variations have been identified for several traits in plants. Some of the major traits includes disease resistance, resistance to the abiotic stress factors like salt stress or heat tolerance or tolerance to the freezing temperatures. Then several other traits related to the health of the plant or the quality of the products like the higher yields, improving the quality of the product, change in the ploidy level of the plants, pigmentation for the ornamental plants which can be of the commercial importance, and so on. In fact, several somaclonal cultivars have also been released for the commercial cultivation. Depending upon the sources of the variation from where they originate, these are grouped in two categories. One is the pre-existing variability and second is the in vitro induced variability. So the first category is the pre-existing variability. The variations that are already present in the explant when we harvest the explants to initiate the in vitro cultures can be categorized as pre-existing variability. In plants, somatic tissues accumulate mutations with the age and differentiation status. More is specialized is the tissue in the plant body, more will be the chances of the mutations present in those tissues. Further, more specialization means these cells are highly differentiated to perform the specialized functions. For example, the leaf versus meristem tissue. Meristem cells are continuously dividing and produce various organs of the plant body. Cells in the meristem divide by normal mitotic divisions and usually uniform genomes are maintained in these tissues. However, the leaf cells are more specialized to perform photosynthesis associated specific functions. And during the process of differentiation for specializing functions, these cells may accumulate more variations. It means highly differentiated tissues have more variations as compared to the less differentiated tissues. The number and amount of variations increases with the age of tissues. That means a leaf tissue that is highly specialized would have less mutations when it is younger as compared to the mature leaf tissue. Even the duplication and endo-reduplication may occur in the specialized somatic cells. That means the specialized somatic cells in a diploid plant may have different ploidy levels. In other words, aneosomaty or polysomatic conditions may arise in the somatic cells. And these conditions are very common in the vegetative parts of the plants. Therefore, chances of having the pre-existing genetic variations in the highly differentiated tissue is higher, and therefore extent of pre-existing variability that is contributed by the explant varies with the source of the tissue or the age of the tissue and health of the explant too. So let's learn a little bit about the polysomaty. Now the polysomaty is the condition where different cells of an organ or different tissues in the plant body may have different ploidy levels, and this condition is called as polysomaty. One of the major reasons for the polysomaty is endo-reduplication of the genetic material in the cell. The term endo-reduplication represents the condition where DNA synthesis cycle takes place within the cell. However, it is not followed by cytokinesis, that is the cell division. The DNA duplication may occur numerous times, and therefore the pyrroid level in the different cells may vary significantly. Endo reduplication is actually an evolutionary strategy to amplify the desirable genes that are required to synthesize the necessary products for the plant growth. It has been suggested to enhance the physiological functions of the cells, as the larger volume will help in tissue expansion and would also help to raise the copy number of the functional genes within those cells where endoreduplication occurs. Endoreduplication usually results in increase in the size of the tissues or the organs. However, the signals that are necessary to change the classical cell cycle to the endoreduplication are still not clear. Endoreduplication events are more common during the cell differentiation process or the cell expansion, and specifically its occurrence is higher in highly specialized cells. Though it is still not clear that if the endoreduplication is genetically programmed or it is just the consequence of the differentiation and environmental stimulation. But certainly various environmental factors like light, temperature, nutritional regimes, or the application of various growth hormones directly influence the degree of endoreduplication. So the overall polyploidization of the somatic tissues may contribute to the somatic or the somaclonal variations significantly if the explants are prepared from these tissues having polyploidy or polysomatic. Second source of these variations is the in vitro induced variations. By now, we have understood the in vitro regeneration process in previous lectures. We select the donor plant and suitable explant to initiate the cultures. The explant is excised from the parent plants and incubated on the suitable nutrient media in the aseptic cultures or aseptic conditions. Any of the genetic variations that are present in the explant tissues when we harvest it from the donor plant is called as pre-existing variability, as we discussed in the last slide. And any variations that will be caused or introduced during the culture now on are called as the in vitro induced variations. So the explant tissues undergo various processes of regeneration like dedifferentiation, redifferentiation to generate the different organs and other morphogenetic responses. Basically, the differentiated cells in the explants are induced to dedifferentiate to make the callus cell. Callus is the mass of undifferentiated cells. Once the enough amount of callus tissue will be formed, these are induced for morphogenetic responses, that is the organogenesis. Then the shoots of suitable size are excised and transferred onto the rooting medium. After the roots develop from these shoots, the plantlets will be ready for their transfer to the soil for hardening. This whole process of in vitro regeneration could be very stressful or a traumatic shock for the living tissues and may result or instigate highly mutagenic processes leading to the mutations or abnormalities in the in vitro regenerated plants. Like when the explant is excised from the parent plant or the donor plant and transferred onto the in vitro culture media, it causes the bonding response, which may be followed up by the sterilization process, which is also stressful for the plant and harsh. Then the induction on the artificial media for dedifferentiation, followed by redifferentiation, or the excision of the shoots and transfer to the rooting media where they are induced for the production of roots. Now all these processes or the changes are very stressful for the living tissues and can lead to the oxidative stress on the growing tissues, which can result in the introduction of genetic mutations. Growth regulators can have significant impact on the introduction of somaclonal variations. The potential of the growth hormones or growth regulators may vary among various growth hormones, and also it can vary among the different plant species. Now, what are the various factors that affect these in vitro induced variations? There may be actually several factors that can result in the accumulation of in vitro induced variations. Majority of these can be categorized in the following groups, that is age of the culture, composition of the culture media, or the growth habit of the tissue, or the inherent sensitivity of the donor plant genotype. The first one is the age of the culture. In the in vitro cultures, tissue is continuously induced for the multiplication at a high rate, and it can affect the genetic stability of the tissues. Generally, the frequency or the rate of the accumulation of variations increases with the increase in the number of multiplication cycles. Further, the time period for which the culture is maintained under in vitro conditions is critical, and longer duration of the culture maintenance increases the frequency of somaclonal variations. Then second is the composition of the culture medium. Now during the regeneration process, right from the explant inoculation or incubation on the culture media to the regeneration of the plantlets, at every stage of the tissue culture, different plant hormones are used. These may be used in the different combinations or at different concentrations. There are several hormones that are used in this in vitro regeneration process, and they are usually grouped in auxins and cytokinin categories, which are most frequently used. And use of these growth hormones is also associated with the accumulation of the somaclonal variations. There are several auxins like naphthalene acid, indole acetic acid, indole butyric acid, or 2,4-D. And similarly, several cytokinins are there like kinetin, vinylaminopurine, or zeatin. And the potency of these hormones to cause the somaclonal variations differs significantly among different hormones. Like 2,4-D has been found to be the most potent for introduction of these somatic variations. Either it induces the polyploidy or selectively favors the division of the polyploid cells. In general, the in vitro morphogenetic responses seems to be dependent on the use of the plant growth hormones or the media composition. However, the genotype of the parent plant also has a significant impact. The regeneration capacity of the plants is regulated at the genetic level. Therefore, the nature of the genotype and their growth habits can influence the frequency of the regeneration, and hence it directly affect the frequency or the capacity for the production of the somaclones too.